Hello my dear students and welcome back to the channel, and today's discussion is for A-level students. So A-level biology students will learn about control of stomatal opening in plants. This topic is from A2 syllabus, and this is from homeostasis in plants, where external environmental factors are likely to influence the opening and closing of stomata in plants. So just like animals, they respond to changes in the external environment and they tend to maintain their internal environment to the constant level. Plants also need to maintain their internal environment to the constant level, and this is important for photosynthesis, this is important for maintaining body temperature, and this is important for maintaining water level in the plant body. So in today's discussion, we are going to discuss that how stomata respond to any change that is occurring in the external environment and how their opening and their closing is controlled. You know that plants do need a continuous supply of carbon dioxide, as it is required as raw material for the process of photosynthesis. And this carbon dioxide enters the leaves through stomata, and it gets accumulated in the air spaces, and from the air spaces mesophyll cells get their required carbon dioxide, and as a result oxygen is produced that is delivered to the air spaces, and from the air spaces air that leaves the stomata that contains high concentration of oxygen and low concentration of carbon dioxide. Stomata need to stay open all the time in order to ensure that during daytime there is a continuous supply of carbon dioxide to facilitate the photosynthesis process on the go and make maximum use of daylight. But this will also result in the loss of water, that water molecules that have evaporated from the cell walls of the mesophyll cells, they have accumulated in the air spaces, and these vapors are likely to be lost when they diffuse out of the leaf through stomata. This transpirational loss of water is also important because it will help in maintaining the plant body temperature. But excessive water loss may cause serious implication. So in order to avoid excessive loss of water, stomata might be closed at time. Even though there is a daylight, there is a continuous demand of carbon dioxide, but stomata might be closed temporarily in order to avoid excessive water loss. Stomata show daily rhythms of opening and closing, and these rhythms are maintained during daytime and even dark times. But remember that opening of stomata is actually controlled by guard cells, and these guard cells are responding to the stimuli. Like during the daytime particularly, stomata are to be kept open. Why? Because there is a low concentration of carbon dioxide in the air spaces. So this low carbon dioxide concentration is the stimulant, and that lets the stomata open during daytime because carbon dioxide is continuously used in photosynthesis. But it has also been observed that due to increased light intensity, again stomata are kept open at a wider diameter. Why? Because increased light intensity will also result in increased rate of photosynthesis, and this will result in more consumption of carbon dioxide, and again it's the carbon dioxide concentration being low that serves as the stimulant. But this opening of stomata also has a consequence, and this consequence is the loss of water vapors through the stomata. Now stomatal closing is also in response to carbon dioxide concentration, but high carbon dioxide concentration. When there is a high carbon dioxide concentration inside the air spaces, particularly the dark time, when there is no photosynthesis taking place, so stomata tend to close. But certain harsh environmental conditions like high environmental temperature, blazing light intensity, dry environment with no humidity at all, or frequently blowing winds may also result in stomatal closure, and this is actually to prevent excessive water loss. But if the stomata are closing during the daytime because of high wind speeds, high light intensity, high temperature, or very dry air around, so this will result reduced photosynthetic yield. Now, how the guard cells are likely to control the opening of stomata? In the cell membrane of the guard cells, there are present ATP powered proton pumps. So these ATP powered proton pumps are in attachment with ATPase enzyme. ATPase enzyme hydrolyzes an ATP molecule, provides energy and phosphate to the ATP powered proton pump. This proton pump moves protons from the cytoplasmic part of the membrane to the outer part of the membrane. Loss of protons and high concentration of negative ions inside the cytoplasm of the guard cells will stimulate the opening of the potassium ion channels present in the cell membrane of the guard cells, and from outer part of the membrane, potassium ion diffuse into the guard cell cytoplasm. This reduces the water potential of the guard cell cytoplasm, and down the water potential gradient from the nearby epidermal cells or the moisture lining, water enters the guard cells. This results in an expansion of the guard cells. An expansion pressure is exerted over the guard cells, and that is more applied towards the outer cell wall compared to the inner cell wall. Outer cell wall is thin and elastic compared to the inner cell wall. So guard cells, when there is an expansion pressure, so they expand outward, and that results in the opening of the stomata. More water enters, the more turgid guard cells are, and more wider the stomata are opened. It has been observed that during water stress condition, abscisic acid, a water stress hormone, is more in concentration in the leaves compared to normal circumstances. Abscisic acid is considered as the water stress hormone. At its high concentrations are observed in the plant parts or plant organs or even in the seeds when there is a very little moisture in them or there is a water shortage. So it has been observed that when there is an excessive water loss being experienced by the plant, or there are certain conditions that account for high water loss. So in that case, abscisic acid concentration is found higher in the guard cells and in the leaves. This abscisic acid actually inhibits ATPase enzyme. So no ATP is hydrolyzed, no phosphate is available to drive proton pumps. So protons are not moved out of the cells in the guard cells. So there is no proton gradient developing, resulting calcium ions diffusing into these guard cell cytoplasm. When calcium ions enter, so negative ions also leave the guard cells, and potassium ion inflow is not only stopped, but certain potassium ions are also allowed to leave the guard cells. This makes the water potential of the guard cells increase, and water down the water potential gradient also leaves the guard cells, making guard cells flaccid. And when the guard cells are plasmolized, they become flaccid. They are dragged inside and they collapse and close the stomata. This is how excessive water loss is prevented. So in a way you can say that when there are water stress conditions around, increased production of abscisic acid makes the guard cells to collapse and close the stomata to prevent excessive water loss. So this is how stomatal opening or stomatal closing is controlled under influence of environmental stimuli, and it is done by the guard cells. Thank you very much, and stay tuned for further updates.